Good morning, Evan, and welcome to Lesson 5. We'll be covering absorption and assimilation. So absorption is a necessary function in the body because everything we put into our body contains nutrients. Those nutrients are needed by the cells in our body, and for that to happen, they need to be absorbed from the food into our body. So where does absorption take place? Most of the absorption. So this is the small intestine. And if you think about it, this makes sense because in the small intestine, the final stage of digestion takes place. Now, in the final stage of digestion, you get the smallest food molecules you could possibly get, and those are then ready to be absorbed. So the small intestine is very, very large. It is 6 meters long. It has transverse waves, villi, and microvilli. All these add to the surface area. So it has a huge length of 6 meters and a huge surface area. The epithelium of the villus, so your top layer, so our epithelium, skin, so the first layer or top layer of the villus is only 1 cell thick. It is richly supplied with mitochondria. Why? Because we need energy for this process. It secretes mucus, which is a carrier fluid. It has microvilli that further increases the surface area, like we said. It is rich with blood and lymph to transport the nutrients, and the villus has muscle tissue. So the villus beats like that. For our bodies to support this movement, they need muscle. So now with absorption, we're going to look at how fats are absorbed. The end product of fat digestion is glycerol and fatty acids. So glycerol is water soluble. It goes straight into the villus, no problem. But your fatty acids are water insoluble. So they first need to attach to bile salts to become water soluble, move through the villus, then the fatty acid and glycerol will find each other again and join. So for instance, you can think about it like you and your friend go to Horwitz Farm for the market. You each have to get there separately and get in separately. But once you're in, you find each other. So once in the villus, and once they have formed fat droplets together, they enter the lacteals. Together with the lymph and in the lacteals, they form a milky substance called chyle. Fat therefore enters the lymphatic system and eventually passes into the blood near the heart. So how are carbohydrates and proteins absorbed? So the end products of these are glucose and amino acids. They are actively carried using the energy supplied once again by mitochondria through the epithelial cells into the blood of the capillaries in the villi. Both are taken in the hepatic portal vein to the liver. Remember, our liver, it sorts out everything good and bad. So these are taken to the liver to be sorted. The blood capillaries in the villi join together eventually to form the hepatic portal vein. This blood vessel carries blood rich with absorbed nutrients to the liver. The blood is then processed, and blood with processed nutrients leaves the liver through the hepatic nerve, enters the heart through the vena cava, and exits the heart through the aorta and goes across the whole body. So it enters the heart through the vena cava and leaves the heart through the aorta. And this is then spread to the whole body. So assimilation. The absorbed nutrients are taken in the blood plasma to all body cells. Assimilation is the using of these nutrient molecules so they become part of the blood. So there's no use having all these good nutrients floating around your body just doing nothing. You want your body to absorb them, use them, and benefit from these nutrients. So how is glucose assimilated? How is glucose used? The hormone insulin allows glucose to enter body cells where various things can happen. So if we look at diabetics, this is the problem with diabetics. They don't have enough insulin in their body to support the amount of glucose needed to be absorbed. So, for example, type 1 diabetics, they need to inject themselves with insulin so that they can have enough glucose absorbed into their body for their body to actually function. So in all body cells, glucose is used for the release of energy during cellular respiration, as we learned in our photosynthesis section. The released energy is stored in our ATP. Oxygen is necessary for this process, and our waste products are carbon dioxide and water. In the liver, the muscle cell's excess glucose is changed into glycogen and stored until needed for cellular respiration. Excess glucose can be changed into fat that is stored in adipose tissue found under the skin. So that is usually coming from our cakes and sweets and all those yummy things. We have put too much glucose into our body. We don't need it. We don't even need it as an excess, as storage in the form of glycogen. So it becomes adipose tissue or fatty tissue. It's stored in there. How are amino acids used or assimilated? In all body cells, amino acids recombine by dehydration synthesis to form a very large variety of proteins, such as enzymes, muscle proteins, and hormones. The type of protein made is determined by the DNA. Surplus amino acids are broken down. So remember, DNA tells us what's going on, where it must happen, what it looks like. DNA controls everything. And if we have too many amino acids, we don't want them, so we want to get rid of them. Assimilation by the liver. The liver converts the excess glucose into insoluble glycogen that it stores. Glycogen is ideal storage because it is osmotically inactive. It is insoluble, water insoluble. The liver reconverts glycogen back into glucose when this is necessary and the glucose is needed. An excess amino acid cannot be stored. It needs to be broken down by deamination. Deamination, you'll look after on the next page, is the removal of an amino group. During this process, the amino acids are converted into urea and more glucose. The amino group, NH2, is broken off from the amino acid, and this forms ammonia, NH2, which combines with carbon dioxide, and then you have urea, which will exit. But the non-nitrogenous base, or part of the amino acid, converts or forms into glycogen, glucose, which will then be converted into glycogen. The liver plays a vital role in the breaking down of harmful substances in our blood, such as alcohol and drugs. Alcohol is broken down by a series of enzymatic or enzyme reactions, and it leaves the body in the form of carbon dioxide and water. Drugs, which include medicinal products, undergo a process called drug metabolism. They will then be excreted in bile or urine. The liver breaks down hormones that the body has no longer need. They have done their job. The liver uses amino acids to make plasma proteins, globulin and albumin. Liver synthesizes vitamin A, retinol, from the provitamin carotene, and it stores fat-soluble vitamins A, D, B12, and it stores iron. So your liver has a lot to do and a lot of work to do. So the more harmful substances you put in your body, the more you're exposing your liver to harmful work almost. It almost hurts a piece of the liver every time it has to go and process harmful substances. So it takes a toll on the liver. So that is assimilation, and we did absorption. We'll be doing homeostatic control of blood sugar in our next lesson next week. But for now and for this week, we are done. So I'll send the activities with this video, and to ask me any questions if you have any, and we'll talk on our Skype session tomorrow, Thursday. But until then, have an amazing day and work hard. Thank you.